Hello and welcome to our third Bio40 Pre-Class lecture. Today we're going to discuss extensions to Mendel's laws. Mendel's laws are a great starting point for understanding genetics, but many traits are more complex than the ones he initially described. So let's look at some situations that build on Mendel's work. We'll explore cases where the concept of dominant versus recessive isn't so clear-cut, where there can be more than two alleles for a certain gene, or a gene can be responsible for multiple phenotypes. Conversely, a single phenotype can be controlled by multiple genes, or even a combination of genes in the environment. We're still dealing with genes as abstract factors now, but understanding these complexities will help us understand the underlying biology. Here's a situation that looks oddly like the blending concept we talked about at the beginning of the course. We start with true breeding red and white flowers, and when we cross them, we get a generation of entirely pink flowers. Neither color appears to be dominant. However, blending would predict that this intermediate phenotype would be stable in the next generation, while genetics tells us that P phenotypes can reappear. And looking at the F2, we indeed see half of this generation has a phenotype of one of the P generation plants. This makes sense based on the genetics we already know. The one twist is that the heterozygote can have a different phenotype than either homozygote. When the heterozygous phenotype is an intermediate of the two homozygous phenotypes, we call this incomplete dominance. In this situation, neither allele can be said to be dominant or recessive to the other. Since each genotype has a unique phenotype, the genotypic and phenotypic ratios will be the same. We can see incomplete dominance in humans with the condition familial hypercholesterolemia. People homozygous for the responsible allele have very high levels of cholesterol in the blood, several times higher than what's common in the general population. But heterozygotes have intermediate levels of cholesterol. Understanding the role of the gene can help make sense of this. The normal gene is responsible for producing a receptor for LDL cholesterol, and the disease allele doesn't produce a functional receptor. With two disease alleles, there's no receptor, so cholesterol levels are very high. One gene making functional receptor can do part of the task, but still doesn't work as well as when people have two normal genes. The other variation on dominance that's important to know about is codominance. Here, the heterozygotes equally express the phenotypes of both parents. The classic example of codominance is AB blood types. The A and B alleles determine what sugar will be present on the surface of red blood cells. Individuals who are AB will equally express both A and B sugars. A homozygous A and a homozygous B individual will have AB children. And two AB individuals will have A, AB, and B offspring in a 1 to 2 to 1 ratio, as we're used to seeing for a monohybrid cross. Again, the genotypic and phenotypic ratios are the same. Blood types get even more complicated because, as you may know, there's also a type O. This is a result of an allele that doesn't put any sugar on the red blood cell surface. Since it doesn't add any sugar, it makes sense that this allele is recessive to both the A and B alleles. So we represent it with a lowercase I, while the A and B alleles are given capital I's with either an A or B following as a superscript. In this case, we have three alleles existing within the population, but it's important to remember that any given individual will only have one or two of these alleles. When there are three alleles in a population, there are 6 different potential genotypes. This chart shows you how the number of potential genotypes scales with the number of alleles. Again, this is only meant to show the number of potential genotypes in the population. Any individual will only have a single genotype with 2 alleles. There's no simple chart for predicting the corresponding number of phenotypes, because that will vary based on which allele pairs have a typical dominant-recessive relationship, which show incomplete dominance, and which are codominant. Moving on, here's another odd case with unexpected phenotypic ratios. When you cross a gray mouse with a yellow mouse, you always get 1/2 gray offspring and 1/2 yellow. It doesn't matter which gray mouse and which yellow, you always get those offspring. Now take any 2 gray mice and cross them, and all the offspring will be gray. They're true breeding. But take 2 yellow mice and you'll see something very odd. The scientists who first tried this examined 1,598 offspring and saw 1,063 yellow mice and 535 gray mice, very close to a 2 to 1 ratio. The appearance of the gray phenotype from yellow parents suggests those parents are heterozygous. But then why a 2 to 1 ratio instead of the 3 to 1 we've seen before? This can be explained if one of the genotypes is not viable. Suppose the yellow allele is dominant to the gray allele, but being homozygous yellow is embryonic lethal. Such mice are never born. The first cross makes sense now. We took a homozygous recessive mouse, crossed it with a heterozygote, and got the predicted 1-to-1 ratio in the offspring. The monohybrid cross works out a little strangely though. When two heterozygotes are crossed, you do indeed see the predicted 1-to-2-to-1 genotypic ratio at conception, but the homozygous dominant mice do not make it to birth. Looking at the mice that survive to birth, you just see the two heterozygotes and the one homozygous recessive mouse, explaining the observed 2-to-1 ratio. Those are the variations on single gene inheritance you should know about. Let's move on to multi-factor inheritance. Many phenotypes are the result of two or more genes interacting. Let's start with a simple example of a novel phenotype that results from two genes interacting. Suppose gene A is responsible for making a red pigment in flowers, and gene B makes a blue pigment. In both cases, a recessive allele means no pigment is made, but one dominant allele is sufficient to get the pigment. When both genes are functional and both pigments made, the combination results in a purple flower. What will happen if we cross two purple flowers that are heterozygous for both genes? Well, we can set up a Punnett square and fill it in like any dihybrid cross. Then we can start assigning phenotypes to each genotype. There are 9 boxes where there is at least one dominant A allele and one dominant B allele. Those flowers will make both pigments and be purple. 3 more boxes have at least one functional A allele but are homozygous recessive for B. Those plants will make red pigment but not blue and therefore be red. Likewise, 3 more boxes have genotypes with a functional B allele but are homozygous recessive for A. These plants will only make blue pigment. Finally, we have one case that is homozygous recessive for both genes and won't produce any pigment. Therefore, we will see a 9-to-3 to 3-to-1 ratio of phenotypes. A real-life example of this kind of interaction can be seen in lentil seed color. True breeding tan lentils are homozygous dominant at one pigment allele, A, and homozygous recessive for another pigment allele, B. Gray lentils are homozygous recessive for A, but dominant at B. When crossed, you get brown lentils, the result of having at least one dominant allele of A and B, and the dihybrid cross gives the 9-to-3 to 3-to-1 ratio we saw before. Here, the double homozygous recessive lentil is green. Because some phenotypes are associated with multiple genotypes, it becomes tricky to predict what will happen if this generation is allowed to self-cross. The green lentils are straightforward enough. They're all homozygous recessive for A and B, and will give rise to only more green lentils. Tan lentils are more complicated. All will be homozygous recessive for B, but only 1/3 will be homozygous dominant at A. These will be true breeding tan lentils. The other 2/3 will be heterozygotes for A and produce a mix of tan and green lentils. Along the same lines, 1/3 of gray lentils will be true breeding, while 2/3 will yield a mix of gray and green lentils. The brown lentils are even more complicated. 1/9 of them will be homozygous dominant for A and B, and therefore true breeding. 2/9 will be heterozygous only at B, allowing their offspring to be brown or tan. Another 2/9, heterozygous only for A, will yield brown or gray offspring. And finally, 4/9 of the brown lentils are dihybrids and capable of producing all 4 colors of offspring, just like the F1. So if we just took all of our gray lentils from the F2 and allowed them to self-cross, what is the phenotypic ratio we should expect in the F3? We will address questions such as this in the next class. And in the next video, we'll cover more of multi-factor inheritance.